Thank you, Brenda, for that lovely introduction. Hello, everyone. I'm Millie Dugal, and I'm excited to be here with you all today. We have two lectures lined up for this afternoon's session. The first is on new trends in trial designs. Leonard Sachs will discuss decentralized trials and the role of digital health technologies in clinical care. The second presentation for this afternoon will be on informed consent. And Mika O'Connell will discuss informed consent and ethical considerations in clinical trials with us. So without further ado, I would like to hand it over to Leonard, and I will be back with you all for the question and answer panel. Thank you.
My name is Leonard Sachs, and I'm going to spend the next half hour discussing innovative approaches to clinical trials. Why bother? Well, the boy in the wheelchair has Duchenne's muscular dystrophy, which, as I'm sure you know, is a devastating X-linked condition affecting the normal production of dystrophin. The condition causes progressive muscular weakness and eventually respiratory failure, with patients usually dying in their late teens or 20s. He's participating in a clinical trial of a new drug, and the trial site is at a pediatric neurology unit in Boston. To get to his trial visits, he has to leave school. His mother has to take time off work and arrange childcare at home. They take a 4-hour flight together to Boston, struggle to get into the Uber in the wheelchair, make their way to the hotel, and the next day they travel to the research institute. They wait for an hour in the waiting room and finally get to the research nurse. She supervises a 6-minute walk test, draws some labs, and does a quick physical exam, and they begin their long trek home. There must be a better way to do this, and that is the focus of my lecture. Technology-enabled clinical trials offer lots of new opportunities. As we all know, there's been enormous progress in communication technologies and telemedicine, which is widely used, in the ability to share healthcare data, images and documents, and the development of biosensors to record physiological features from patients. There are also opportunities for remote data acquisition in clinical trials, which are remarkable. They include getting data from participants, getting data from healthcare providers, and even from healthcare institutions. In September this year, we published our final guidance on conducting clinical trials with decentralized elements. Decentralized elements allow trial-related activities to occur at locations other than traditional clinical trial sites that are convenient for trial participants. This can be, for example, in patients' homes or local clinics and facilities. Why are regulators interested in decentralized trials? Well, first of all, accessibility. These trials provide accessibility for patients with rare diseases who couldn't get to trial sites. They provide accessibility to patients with mobility or cognitive challenges, and they provide accessibility to a diversity of participants from different socioeconomic or cultural backgrounds who formerly did not participate much in clinical trials. They improve convenience for patients. They provide other efficiencies. For example, they save on travel, on the need for physical facilities when activities take place at patients' homes or local clinics. And they also take advantage of community healthcare providers. And of course, we can avoid the transmission of contagious diseases such as we saw with COVID. Decentralized trial procedures are not new. In outpatient trials, once participants leave the study site, many trial-related activities take place remotely. For example, there is self-administration of investigational products. Adverse events occur in patients while they're at home and they present to local clinics or emergency rooms. Patient diaries have been used for years to document certain symptoms, such as seizures, such as pain, etc. Interactive voice response systems are traditionally used in many clinical trials. Telephone follow-up is often a feature of long-term follow-up in a trial, and electronic informed consent is fairly widely used since we published a guidance on this in 2016. Decentralized clinical trials involve a bunch of different strategies to track trial activities to the patient. In the middle, you can see a participant. The woman has paroxysmal atrial tachycardia, and you can see on the left she has remote telehealth visits with the investigator. She signed her electronic informed consent electronically. Her drug is delivered to her home. She goes to the local clinic for her blood work. She goes to the local healthcare provider for a clinical examination. And you can see the last example is a digital health technology that she wears. It's a mobile cardiac monitor that reports episodes of atrial tachycardia. Let's talk a little bit about remote trial visits. These are trial visits where the investigator can supervise remotely. And these are commonplace in telehealth, but they have certain challenges. For example, they have to be in line with local regulations on telemedicine. You can't do a physical examination over the phone. Video photography may not fully capture the features of a lesion if that's what you're using. Patient engagement in the absence of in-person contact may be a challenge. Complex drug administration procedures are difficult to do over the phone or over a video call. And in situations where you need close medical supervision, e.g. for infusion reactions, having the patient do the visit remotely is probably not suitable. What about the use of local healthcare providers and facilities? There are resources and qualified healthcare providers in the clinical care environment who may be able to be used in clinical trials. And there can be delegation of routine clinical activities to patients' local clinics or to healthcare providers for routine clinical procedures, for example, x-rays, clinical examinations, lab tests. It's important to ensure that providers have the appropriate qualifications, and it's important for investigators to review the data regularly to make sure that it's complete and correct. Home visits are a novel approach. They may either rely on dedicated trial staff or contracted healthcare providers going to patients' homes to do certain trial-related activities. Mobile trial units in vans are currently being developed by many sponsors, and this is another way of getting trial activities to the patient, although obviously the perimeter is limited. And all these approaches are designed to extend the physical reach of the trial. Investigational products can be directly distributed to patients. It's important for the investigator to control the release of the product to the trial participants. Local state laws differ on direct distribution to patients. They may require locally licensed healthcare professionals or pharmacists, and this should be clarified before the trial is underway. Packing and handling are important considerations to make sure that the product arrives in good order, and there have to be plans for patients to dispose of unused product. Electronic informed consent, as I mentioned, has been referred to in a previous guidance published in 2016. It's already in widespread use. It allows patients to review and sign informed consent at home. It may provide videos and graphics to make the process more informative and more easily understood. But of course, remember that signed consent must be obtained before enrollment. When considering whether an investigational product is suitable for decentralized clinical trials, the nature of the drug should be considered to determine whether administration outside of a clinical trial site is, as part of a DCT is appropriate. One should take into account the complexity of administration, the safety profile of the drug, and the stage of drug development. Drugs in early development are probably less well understood and probably not suitable for administration in a decentralized way. It may be appropriate for local healthcare providers or trial personnel working remotely to administer the investigational product at local healthcare facilities or at participants' homes. Remember that hybrid DCTs using both local and central visits may be considered for drugs that require supervised but infrequent administration. Investigator oversight. Investigators have several responsibilities. Firstly, oversight of the trials. They have to ensure that data provided by local healthcare providers is reviewed on a regular basis. Investigators should make sure that the data they receive are complete and consistent with the requirements of the protocol. And it's the investigators who will need to make decisions about whether a patient's complaint to a local healthcare provider represents an adverse event, whether a finding reported by a local healthcare provider represents a study endpoint, or if a protocol-directed change in treatment is necessary, because it's the investigators who are familiar with the clinical protocol. As far as patient retention goes, when patients are seen remotely, the jury is still out. We don't know whether patients are less likely to follow up, and this is something that we're anxious to find out. Investigators are also responsible for ensuring data privacy. A couple of thoughts about safety assessments in a decentralized clinical trial. Trial participants must be able to contact trial personnel to report adverse events and to have pertinent questions answered. Trial participants should be able to arrange for unscheduled visits using telehealth or an in-person visit as appropriate. If significant safety risks emerge because of remote administration or use of an investigational product, sponsors must discontinue the remote administration or use. And if authorized in the protocol, routine safety monitoring involving lab testing and imaging may be performed using local clinical lab facilities. Protocols should specify how adverse events will be identified remotely and will be evaluated and managed, and how care will be provided for adverse events that require urgent or in-person attention. Sponsors also have responsibilities in decentralized clinical trials. These responsibilities are the same for DCTs and for traditional site-based trials. Sponsors should strive for diversity and inclusiveness in trial populations. Sponsors need to account for multiple sources of data collection in a DCT and the data management plan, and they should describe in the trial protocol how the operational aspects of the DCT will be implemented. Some important considerations for decentralized trials are firstly that not everything can be accomplished remotely. Many decentralized trials will end up as hybrids, combinations of remote visits and visits to investigator sites when procedures such as detailed physical exams and other in-person activities are required by the protocol. To turn to a different sort of trial, this is a guidance that we've just published called Integrating Randomized Controlled Trials for Drug and Biologic Products into Routine Clinical Practice. These are trials which are sometimes known as point-of-care trials or pragmatic trials, and the idea is to conduct the trial in a clinical practice setting where the patient normally gets their medical care. The medical literature abounds with these sorts of trials, point-of-care trials, pragmatic and large simple trials, and what all these trials have in common is they rely on integration of clinical research with clinical care. Now, these approaches have not been significantly adopted for regulatory submissions. A poster child for one of the trials conducted in clinical practice was the recovery trial, which was done in the UK for COVID-19. This trial reportedly recruited 40,000 COVID patients through the National Health System in the UK within 6 weeks, and they were able to show the mortality benefit of steroids, tocilizumab and baricitinib in treating patients who were hospitalized with COVID. So practice settings allow for engagement of large numbers of patients in short periods of time, and these trials reflect the effectiveness of treatment in real-world environments. Accessibility of clinical trials to patients who wouldn't normally participate is another feature of these trials. To integrate randomized controlled trials into clinical practice and to conduct them where patients get their routine care, it's important that trial designs and activities are streamlined to align with clinical practice. These trials leverage established healthcare institutions and existing clinical expertise in the medical community to reduce startup times, speed up enrollment, and improve accessibility and convenience for patients. Real-world data from electronic or other healthcare records may be used in these trials. Trial-related activities may be conducted as part of routine practice with participation of local healthcare providers in these healthcare institutions, and dedicated trial staff may participate if needed to perform activities that require research-specific expertise. As mentioned earlier on, digital health technologies may also play an important role in decentralized clinical trials. In the context of clinical trials, we are interested in digital health technologies such as wearables, interactive applications, and instruments placed in the patient's immediate environment that measure a clinical feature of interest in a clinical trial. These may be instruments like smartwatches, cell phones, mobile glucose monitors, etc. This again is a child with Duchenne's muscular dystrophy, and he's doing the 6-minute walk test to evaluate drug efficacy. This is a clumsy test. He has to walk as far as he can within the space of 6 minutes. What if he has a sore foot or had a bad night's sleep, or if the supervisor of the test motivates some kids more than others? There must be better ways to measure this. This picture is of an accelerometer. And accelerometers are versatile tools for Duchenne's and many other diseases. Accelerometers are used all over. They're present in our cell phones, smartwatches, and Fitbits. Triaxial accelerometers give good three-dimensional pictures of movement. They can be very helpful in measuring steps or other activities taken by a Duchenne's patient. There's no need to depend on snapshot measurements and clumsy tests like the 6-minute walk test. Measurements can be recorded over long periods of time, so we don't rely on single measurements, and comparisons with baseline performance can be made. Accelerometers are not the only sort of digital health technology. Here's an example of several. For example, galvanometers, which measure electrical current, may be used. The output of these is typically voltage, current, or impedance, and they may be used, for example, to measure heart rhythm. There's an interpretive algorithm, and the combination of the algorithm that measures heart rate and the signal from the galvanometer results in a DHT. This would be a mobile cardiac monitor. The same would apply to accelerometers, which have algorithms to measure walking, scratching, sleeping, or tremor. Photoelectric cells, which can be used to measure blood oxygen saturation, electrochemical sensors, and a good example are continuous blood glucose monitors, which again rely on algorithms and calibration curves to make frequent measurements of blood glucose, thermocouples, which are used to measure temperature, etc. As far as these biosensors go, they can measure clinical features, some of which may be discrete events like steps, breaths, coughs, pulse beats, seizures, tremor, or forced expiratory volume. On the other hand, they may make continuous readings like glucose reading, continuous glucose readings, pulse oximetry, temperature measurement, ECGs, electrocardiograms, and blood pressure. It's worth reminding ourselves of some of the novel types of data that biosensors can provide. Firstly, they can provide rich continuous data instead of snapshots. So, for example, we can get the average number of steps a patient takes per day versus the 6-minute walk distance. We can get continuous glucose monitoring versus a static hemoglobin A1C, and that way we can monitor hyperglycemia and hypoglycemia and time in range and so on. DHTs give us the ability to detect rare events, for example, things like falls, arrhythmias, seizures, or apneic spells, and these often don't occur during a particular study visit. We can get data from patients who cannot respond. For example, we can measure the scratching in infants at night with atopic dermatitis. We can measure sleep in patients with dementia. We can get dose response information. For example, we can capture the on-off effects of Parkinson's drugs. We can make new types of measurements that were not possible when we saw patients in the clinic. For example, we can measure gait stability that may predict falls. We may be able to measure the amount of coughing that a patient does, sneezing, tremor. We can measure behavior patterns in dementia or in depression. And another important feature of digital health technology is that they may allow us to detect functional abnormalities very early. For example, coordination abnormalities, gait abnormalities, or reaction time. And these may be very important in, for example, neurodegenerative conditions where we want patients to be treated before their conditions advance too far. In order to rely on digital health technologies and biosensors, verification is an important process that confirms that the sensor is working as needed. And here you can see an example of a shake table that's used to test the accuracy of accelerometers to measure acceleration. On the right-hand side, you can see the performance of an accelerometer at different temperatures. An important study that's needed to give us confidence in digital health technologies is an analytic validation study. This confirms that the interpretive algorithm is working properly. And here you can see on the left-hand side the raw signal from a triaxial accelerometer, both for subjects running and subjects walking. And you can see roughly the deviations that correspond with steps. And on the right-hand side, you can see the ground truth recorded in a video recording. The video recording is scored by an observer. The interpretive algorithm gives the step count in 5 minutes at 480 with the accelerometer, and the observer reports a step count of 477 in 5 minutes. And there is just a theoretical example of how we can calculate the sensitivity and the specificity of the interpretive algorithm for measuring steps. It's important to consider confounders of the measurement as well when we rely on digital health technology. Is the result affected by how the patient wears or uses the DHT? Are there things that the patient might do that would be misinterpreted by the DHT, for example, tapping a foot or riding a bike? As part of validation, one of the questions is whether the DHT is suitable for use in the trial. This is an operational issue. Is the DHT ugly or elegant? Is it easy to put on? Is it easy to operate? Is it comfortable to wear for the time that you need to wear it? Is the battery life long enough? Is syncing data a simple process? And is it appropriate for patients to bring their own devices, for example, their own smartwatches or cell phones? Many of the DHTs that I discussed before are passive measures. They measure the patient without having the patient actually do anything other than carrying on with their normal functions. But interactive DHTs are another promising possibility for measuring clinical features or clinical performance. And here you can see examples of this. Probably in its most pedestrian form, a patient-reported outcome on a cell phone is one form of DHT. A cell phone camera can be used actively by the patients to capture a lesion. Vision tests can be done on a cell phone. Coordination tests can be done on a mobile app. This is an example of an app where there are 2 buttons on the screen and the patient has to press them alternatively as many times as they can within a given time interval. Mobile spirometers are another thing that can be used interactively. And finally, there are other more elaborate machines that can be used to measure patient strength or patient activity. You can see examples here of a portable fixed dynamometer, which is used to measure muscle strength. Now, it's important to distinguish between the tool that's used to make the measurement of a clinical feature versus a clinical endpoint that makes use of the clinical feature to evaluate a drug effect. Verification and validation that I mentioned earlier are technological assessments, and they only address how well the technology measures the clinical feature of interest. On the other hand, justification of an endpoint or clinical outcome assessment is a clinical issue. It addresses where the clinical feature is a meaningful way to assess the response to treatment, and it has nothing to do with the instrument that's used to make the measurement. This is an example of a clinical outcome assessment that was qualified by the European Medicines Agency. This is the 95th centile of the stride velocity, which was qualified as a primary endpoint in studies in ambulatory Duchenne's muscular dystrophy. What it represents is the top 5% fastest strides a patient spontaneously takes in the normal daily environment over a predefined period, say a week or whatever. And remember that this is device agnostic. It doesn't matter whether this is measured by a video camera or whether it's measured by an accelerometer. Developing endpoints that are based on digital health technology measurements, it's very important to determine that these are clinically meaningful measures of drug effect. The following are considerations when you formulate the endpoint. What exactly is being measured? In our example, this would be something like steps. What is the time window of observation? The trial may be a trial over 4 weeks. And what is the formula to calculate the response in each patient? In this example, this would be a change from week 1 to week 4 in the average daily step count. And then other considerations are whether the endpoint is a clinically meaningful measurement of drug effect, and this relies on comparison with existing benchmarks of performance. One example would be the Unified Parkinson's Disease Rating Scale in the case of patients with Parkinson's. Sometimes we may rely on other patient-reported outcomes or the 6-minute walk distance as prior benchmarks of drug activity. And it's very important to include the input from patients, from caregivers, professional societies, disease experts, and regulators when deciding whether you've established a clinically meaningful endpoint that's measured by a DHT. I included this slide just to give you an idea of the types of endpoints in general that are used in clinical trials, in registrational trials. This is a summary of those that we've seen between 2015 and 2020. And you can see that there are different types of endpoints. About 30% of trials rely on biomarkers, things like chemistry measurements, hemoglobin A1C, hematology measurements, for example, neutropenia, pathology measurements, microbiology measurements, for example, sustained virological response in HIV or plasma viral in hepatitis and plasma viral load in HIV. Imaging is another important feature often used in cancer trials, and other examples include bone mineral density, spleen volume, and then physiological or functional measurements were the endpoints in 7% of the trials we looked at. These are things like the 6-minute walk distance, which we've described, detection of normal sinus rhythm, forced expiratory volume, and sleep studies. And then 22% of our trials during that time period relied on clinical events or clinical signs, for example, death, hospitalization, major adverse cardiac events, or multiple sclerosis relapses. And finally, 32% relied on clinician-reported outcomes or patient-reported outcomes. And some of these examples are clinical scores. For example, the Toronto Western Spasmodic Torticollis was a rating scale that was used. The Hamilton Depression Rating Scale for depression, rheumatology scales, ankylosing spondylitis scales, psoriasis severity indexes, and so on. Based on that list, it's obvious that there are certain types of endpoints where mobile technology tools may play a role. They can play a role in clinical laboratory measurements, as we know, measuring things like continuous glucose monitoring, pulse oximetry. They can make physiological measurements of heart rate and rhythm, breathing and lung function, seizures, syncope, temperature, weight, sleep. They can make measures of clinical events, which I've mentioned before, seizures, falls, arrhythmias, apnea, spells, and they can be used to make performance assays, for example, to assay stamina, strength, coordination, abnormal movements, and cognition. DHTs can be used in different ways in a clinical trial. They can be used for enrollment screening, and they can be used to enrich certain populations, for example, those with severe functional abnormalities. They help us quantify disease severity, and they may be able to establish the functional status at enrollment. They can be used to monitor safety, for example, the identification of rare adverse events, and they may provide real-time access to safety data. They can be used to visualize the dose effect, and they can be used for clinical trial endpoints, which we've already discussed. As far as endpoints go, the use of DHTs would be most compelling in superiority studies, where one arm of the study is shown to do better than the other arm. Our concern with non-inferiority studies is that if the DHT performs badly, it may make both arms look the same, and we may not be confident that the drug has any effect at all. Points on our regulatory position: digital health technologies and clinical trials generally do not need to be approved or cleared by FDA for marketing unless they are being used as medical devices. Our regulatory standards are the qualities of the data from the DHT adequate to provide substantial evidence of effectiveness when testing a drug? Are the data attributable to the patient? Are there processes in place to ensure data integrity, data security in transit and during storage? These are just data quality issues which our regulations address as far as electronic systems go. Brings me to my concluding slide. Electronic technology provides many new opportunities to exchange information, to obtain clinical data remotely from patients, from healthcare providers, from clinical institutions, and to make improved clinical measurements. Interoperative electronic systems will support integration of clinical care and clinical research, and these advances will play an increasing role in improving trial efficiencies, convenience for patients, and access for diverse participants, including those with rare diseases. If you're considering an innovative trial design for a drug-specific application, you should contact the relevant review division for a pre-IND meeting. For engagement on digital health technologies or decentralized clinical trial designs that are not related to a specific drug development program, you can contact us at DHTs for drug development at fda.hhs.gov, and we will determine the best forum to address this. Questions? All all digital health technologies for use in clinical trials need to be cleared by or approved by FDA, true or false? Well, that's false. Only those that are being used as medical devices need to be cleared or approved. Justification of an endpoint as meaningful depends on the DHT that's being used to make the measurement, true or false? That's false. Justification of an endpoint is independent of the method of measurement. And finally, trial complexity is not a concern when trials are integrated into clinical practice, true or false? And that again is false. Trial complexity is a concern. Trials integrated in clinical practice should generally be streamlined with simple procedures that align with clinical practice. And I'll end there. Thank you.